Okay, good morning, everyone. To introduce myself, I'm Gary Firestone. I'll be with you for the last third of the semester. I've been in the department here on campus for about 30 years. It's my 31st year, and about my 10th or 12th year, something like that, in Bio 1A. So I've been doing this for a little while anyway. Give me a sense of you guys. How many of you are intended MCB majors? Okay, intended IB majors? Good. Intended public health? Intended some other science that I didn't mention? ESPM? Is that one of them? What else you guys? Plant biology, plant microbiology? Yeah, okay. Any of you social science majors? Okay, humanities majors? You're the brave ones. You're the brave ones. Okay. A little bit of an introduction. I hope you guys have sufficiently recovered from the exam and from Cal's women's basketball team's very close game last night. That was not fun to watch at the end. Mike tells me the exams will be ready in your study sections, where those are later in the week. And so that's what he told me. Anyway, to give you an overview of what we'll be talking about for the last third of the class, we'll do tissues and cells today. We'll spend a couple lectures on hormone action, human reproduction, and stem cells and development. Systems fly by. There's about 6 systems I'm going to do in the last 5 lectures or so. The common theme is hormone signaling and functional connections. So there'll be a common thread. That's why I do hormones first. And I will stress the human biology side of it as much as I can. Okay, so there's the 2 common threads there. So in terms of today, have any of you gotten a hold of a reader? Be able to buy the reader? It's available. Okay. For those of you that don't want to pay for it, we'll be sending it out on BSpace. Mike, did you send out the first couple? Okay. And then the whole full PDF will be available Wednesday for those of you who want to just print it yourself. Although probably the cost of printing it is probably greater than the cost of the actual manual, but it's your decision on that one. So in terms of what we're going to do today, I will refer to the reader quite a bit. In terms of my part of the class, the only thing I'll be responsible for is what I mention in class. Okay? If there's something in the book that's different, it's fun reading, it's worthwhile to read, gives you a different perspective, you won't be tested on it. Same thing with the reader. There's a few extra things in the reader. In addition, there's a few things I'll mention in class that are not in the reader. So the reader's designed for you to embellish what's written there with what I actually say in class. But it has pretty much all the basic concepts that I'll be talking about. I will be following it quite carefully. Okay? So we'll do today is hierarchical organization briefly. Then we'll talk about cell-cell interactions. Then we'll get into the 4 types of tissues: epithelial, connective, nervous, muscle, organs, if we get that far. And at the end of each section in the manual, there's a set of questions that you should at least attempt to answer. It'll give you a sense of what you know and don't know about the class. For me, I have office hours Monday noon to 1, which I think is the same as the previous faculty had, right? Monday noon to 1, and also Thursdays 2 to 3. In addition, if you have questions by email, that's totally fine. Gives me a sense of where you guys are at. So if something comes up, ask a question. And if it's an interesting enough question, or if enough of you ask the same type of question, I'll go over it in class. The only thing I won't accept by email is, could you please reproduce lectures 2, 5, and 7? Okay, something like that. That's out of bounds in my opinion. Okay, and so forth. So any questions about what I just mentioned? Any questions at all? Okay, all right, good. So the way I like to introduce this part of the course is by hierarchical organization, and that is molecules to cells to tissues to organs. They're all interconnected. And the number you don't need to know, you have about 50 trillion cells, and there's about 200 different cell types. And cells are sort of the basic unit of life. They're formed by mitosis. 1 cell gives you 2 cells. By fusion, 2 cells come together, gives you 1 cell, such as the formation of a zygote, which we'll talk about in human reproduction. And with new technology, you can take a nucleus, throw it into an enucleated cell, and get a live cell out of it. So this is a definition of a live cell that probably did not exist about 20 years ago. Certainly when I was your age, it didn't exist. And these cells regulate the tissues and organs and are regulated and regulate molecules. So the molecules in a cell give you specialization, function, and differentiation. So the DNA, your DNA of a stomach cell is the same as an intestinal cell, but the functions are quite different. It's because of what genes arise in those cells and who they interact with. These cells then have to come together and functionally coordinate in the tissues, and the tissues functionally coordinate into organs, organs functionally coordinate into organ systems. This is all very important because in a disease state, you can be defective in a molecule that then affects the function of a cell, that then affects the functional coordination of a tissue and organs and so forth. Secondly, certainly in the case of cancer and some other kinds of disease states, you can have changes in functional coordination of tissues, which then goes back and affects how a cell functions and which molecules are ultimately produced or how they function in a cell. And that's all important to think about. Ways you can manipulate this is through hormones and various signals, and the reason why I hit the hormones is I'm a firm believer that you are your hormones, and as a result, you can change how a cell functions, change tissue coordination, change the molecules in a cell. And you guys will probably see this later on in terms of, many of you like going into research or medicine, probably a lot of you might want to do that. Okay. It's the beginning of it now, but you'll probably see a lot of it later on. In gene therapy, you say, okay, we're missing a gene. Let's give that gene to that person, and we cure everything. However, if you give a gene to a cell, i.e. adding a molecule, you may or may not get the correct tissue functional coordination, depending on when you give the gene, and therefore not the correct organ system. So it's not just applying a gene to a cell, but it's also making sure everybody coordinates. Everybody has a functional coordination. And this will be sort of the bottom line theme for all these lectures, that everything comes together. And my point of view is of cells and how you functionally change a cell, and therefore how you coordinate to the tissues and organs, and then down to the molecules. I'm sure you've had a lot of molecules up to this point in the class. Okay? Questions on that? This is just sort of an overview of this. And again, please do not memorize that there are 50 trillion cells. I will not ask you that question. Okay? It's a nice little tidbit of information, but that's about it. Okay. So what we're going to do today is talk about how cells come together to coordinate the tissues. And one of the ways that happens is through cell-cell interactions. Oops. It's very important. Because when cells come together, here's a tissue, and here's a bunch of cells. They come together in a tissue, they have to talk to each other. They have to be regulated. And one of the ways in which these guys talk to each other is through communication. They communicate. And they do this through a bunch of pores in between the cells called gap junctions. And I'll put this up here. And so there's literally pores in between most of your cells. And these cells, almost all cells have them. Examples of cells, almost all cells. And exceptions are that we'll talk about are free swimming or free living cells, such as red blood cells, RBCs, and spermatozoa. Are two examples of cells we'll talk about that don't particularly have a lot of gap junctions, because there's nothing to coordinate in that sense. So let's think about a gap junction here. It's a huge pore. Not really huge. I'm drawing it huge. And these are the 2 cell types that come together. And they allow for the movement of molecules in between the cells, so they can coordinate functions. And what the coordination is they allow these cells to be synchronized. They allow tissues to be synchronized. Can any of you think of anything going on right now in your bodies that have to be synchronized? If I had to say to you synchronization of a tissue, because if it doesn't synchronize, you're not going to be around much longer. Can you think of anything? Yeah, nervous tissue, what else? Yeah, like your heartbeat. So what makes all the heart beat contract at the same time is because there's gap junctions between all the cardiac cells. It makes them all go around the same time. So heart contractions. And one we'll talk about with human reproduction, contractions during labor are all synchronized because of gap junctions. Now these pores allow small molecules to go through. They're not gaping holes. It's more like that leak on the ceiling here. It's small molecules can go in between. So things like ions, amino acids, lipids, sugars, etc. Small cell signaling agents can get through on gap junctions. So it's not like these cells don't talk to each other. They have to talk to each other to be functionally coordinated. Okay? Now the gap junction itself, these gap junctions are comprised of 6 proteins on either side of the cell. Here's the plasma membrane. Close enough. 5 rounds up to 6. And these different proteins, so this is the plasma membrane between the cells, and each of these proteins are called connexins. And there's different combinations of connexins in different tissue. Those details are not important. And there's also defects. For example, a lot of times in cancer, there's a dysregulation of some of this. And what happens is sometimes the expression of these connexins goes down, and therefore you don't have the right number of gap junctions or functional gap junctions. The cells can't coordinate with each other. So a lot of times in a cancer cell, even though it's growing without any regulation, it also is missing a lot of critical cell-cell interactions at the same time. Okay? And each cell, here's cell 2, cell number 1, each of these supplies 6 connexins that come together. So they form a pore. So the pore has a cell 1 and a cell 2 portal, so to speak. And they come together, and they recognize each other, and they bind, and they form this pore. Okay? But this is happening all over the place. When we talk about reproduction, there are gap junctions between the follicle and granulosa cells. You don't need to know what that means at the moment, but we'll talk about it. That coordinates the development of the follicle, for example. So everything is coordinated through these gap junctions, or one aspect of coordination. Okay? Questions on gap junctions? Pores, gaps, pores, easy to remember. Okay? Now, besides, you can also, when you're in between the cells, you can also have adhesion. And there's several ways which cells can contact each other, called cell-cell contact. One of these is through some molecules called adherence junctions, which is very strong adhesion. So these are belts of adhesion. Remember, cells are three-dimensional. So picture a belt, right? So anywhere across here, if there's another cell right here, I can have contact with that cell, and therefore adhesion. These are very strongest kind of cell-cell interactions. They can bind anywhere around each other. And adherence junctions have a structure that will be similar to the rest of the junctions we'll talk about. So if here's cell number 1, it has a transmembrane protein. Let me over here. A transmembrane protein called a cadherin. There's a bunch of cadherins. You don't need to know the differences between all the different cadherins. Just know this is the main transmembrane protein. Here's the plasma membrane. The main transmembrane protein that allows for cell-cell adhesion. The other cell, cell number 2, also has cadherins. Okay, here's cell number 2. And these cadherins will bind to each other. In addition, there's a whole bunch of other proteins on the intracellular side. So it's a protein complex. It's very important to appreciate. There's a large protein complex that forms. One of the proteins that are involved in it is very important. There's several of these called catenins. And the catenins are regulatory molecules, and they also interface with the cytoskeleton. You guys had the cytoskeleton a little earlier in the class? Yes? No? Yeah? Like actin and all this kind of good stuff. But these guys connect up with the cytoplasm. So what happens is you form this intracellular membrane complex that then functionally makes these cadherins bind to each other, and you have this very strong belt of adhesion around the cells. Okay? One area, so you can imagine, if you have a dysfunction in this, well, you have a dysfunction, say you don't have a lot of cadherin or catenin around, you'd be defective in what? Cell-cell contact, right? Or changes in that. And a very good example of that is metastasis of cancer cells. A cancer cell has to come off the tumor, go somewhere else in the body, and attach somewhere else. Part of that is allowed by changes in these cadherins and catenin molecules that allow for detachment and then reattachment somewhere else. And there's some thought that this is one of the areas you could target for anti-metastasis drugs, is targeting adhesion junctions. Okay, very important. Any questions? Cadherins is the name of the protein. These are the proteins. Adherence junction is the name of this entire complex. So this is an adherence junction. Adherence adhesion. That's probably where the derivation of the name comes from. An adhesion junction, very good question. Adhesion junction has not only the cadherins, which is the transmembrane protein. This is the plasma membrane. PM is plasma membrane. Okay? Also has these intracellular proteins that help regulate it. Okay? Other questions? Catenins, you don't really need to know this that much, but catenins regulate the cadherin from working. Okay? It's just there's an intracellular regulators. It's a very complex and interesting complex that forms. But it plays a role in how well the cadherins will function. So if you knock out a catenin, this cadherin is not going to do much. Okay? Yeah, these guys always, you don't have to know this, these interconnect to the cell, inside the cell. And that helps stabilizes it. I'm doing this because the light's right in my eye here. I can't see the rest of the question. Okay? Very good question. What way does it regulate? Don't need to know that. You don't need to know that. There's entire courses on these kinds of cell-cell interactions. But part of it changes, there's phosphorylation changes, the signals come in and changes the conformation of the cadherin, therefore it doesn't quite interact as much. Don't need to know. Some details you don't need to know. Okay? Now, there's another kind of adhesion complex called desmosomes. Desmosomes are spots of adhesion on a cell. Okay? So these are very weaker, much weaker kinds of binding. And the structure has some similarities to what we just talked about. Here's cell number 1. And there's a lot of detail you don't need to know, but for now we'll call these cadherin-like molecules. They're in the cadherin family, but they're slightly different for the desmosomes. In addition, there's a bunch of proteins on the inside of the cell. Here's the cell number 2, same thing. And these proteins on the other cell are a bunch of other proteins. As an example is desmoplakin. So if desmoplakin in a desmosome binds to a cadherin in an adherence junction, it's not functional. Okay? The desmoplackins, for example, have to bind to these cadherin-like molecules in desmosomes. These are spots of contact. Okay? Very similar, but with some interesting differences. I'll put this back up in a second. There is a very interesting syndrome. So if you have defects in desmosomes, you'd have defects in what? Kind of contact between cells, but not strong contacts, right? And there's an example in skin cells. I'll put this right here. We have a defect in the desmosomes and allows water movement in between the skin at the skin cells. And what this gives you is a disease called a blistering disease. Okay? So here's, even though this is not strong contact in the skin, it's important. You don't need strong contact in the skin. You need lighter contact or weaker contacts because your skin normally sloughs off anyway. But if you have a defect in desmosomes, you get a blistering disease because it allows too much water to get in there. And this is a syndrome you guys may see, those of you going on to med school later on, will probably see examples of that. It's not that uncommon, or different forms of it are not that uncommon. Okay? Questions on junctions that allow contact? There's one more we're going to talk about in a second. Movement, sorry. Water moves, how's that? Make it a little shorter, okay? Now the last kind of, and we'll see how this works together in epithelial tissue in a minute. You also have tight junctions, which involves sealing and selective movement in between the cells. An example is if you have a jar, a glass jar, right, and you have water, you take the lid off, what's inside the lid? It's usually a rubber gasket, right? So if you put the water in, put the cap on with the gasket, tighten it, turn it upside down, what happens to the water? It stays in the jar, right? But once you take the lid off, take the rubber gasket off, put it back on the jar, and then turn it upside down, shake it a little bit, what usually happens? Water starts dripping out, right? So that gasket is the tight junction. The lid would be the adherence junction. So the adherence junction provides contact. The tight junction provides sealing, like a rubber gasket or seal. We'll talk a bit more about this, but the structure is similar to what we just talked about. It's a common theme here. What's emerging? You have a transmembrane protein. These are called occludins. Claudins, there's a family of a lot of them. And they form a contact with the same group on the other cell. Here's cell number 2, cell number 1. And you have a whole bunch of you have a whole bunch of intracellular surface membrane proteins. Again, to form a protein complex. One of these is called ZO1, zonal occludins 1, you don't need to know the name. ZO1 regulates how the claudins and occludins come together, and it's a focal point of cell signaling target. It's a target of a lot of cell signaling that you'll see, may or may not see later on. But the point is, all these structures, desmosomes, adherence junctions, and tight junctions, all have a transmembrane protein with an intracellular protein complex that forms on the inside of the membrane. It's the transmembrane protein that provides the sealing of the contact, and it's the proteins in the inside of the cell provide the regulation. And you can see that common theme there. And there are a lot of disease states that have changes in tight junctions. Cancer is one for sure. Another one is called multiple organ dysfunction. And a lot of times when your organ systems start breaking down, one of the things that breaks down are the tight junctions. And we'll see in a minute here as an example, like the intestinal cells. You can't take in nutrients very well because of this. And you also have glaucoma. This disease is related to changes in the regulation of sealing or tight junctions. Right? For those of you who go for eye tests, it's probably not critical for you guys, it's only my age. You go for those pressure tests on your eyes. Well, the tight junctions allow for pressure or not. Regulates that pressure on your eyes. And people with glaucoma, it's been tracked to some changes in tight junctions. Okay? Questions on that? Gap junctions, adherence junctions, desmosomes, tight junctions. Okay, now you know almost all the key ways cells can regulate each other in a tissue. All this is going on. Any questions? Yes? Oh, very good question. Outstanding question. Are they together? And the answer is yes, in epithelial tissue, which is exactly the next topic. So thank you for the segue. I appreciate that. Okay? So, yeah. Yes. This and this are the same. Cell 1 and cell 2. Okay? Other questions before we go on here? Okay. Outstanding question. Do they all work together? And the answer is all of these work together. Cells can have gap junctions, adherence junctions, and tight junctions, all in the same kind of interaction. Okay? So there's a lot of functional coordination. But the adherence and tight junctions work together very well in epithelial tissue. So there's 4 kinds of tissues. Epithelial, connective, nervous, and muscle. And in epithelial tissue, basically, the way it's defined as is cells. They grow on a basement membrane. Basement membranes are made of fibers, oligosaccharides, and so forth. And they sit down on these basement membranes. And epithelial tissue has lots of functions, but the 3 that I want you guys to think about are protection. Epithelial cells surround all the organs in your body, and they protect from stress, environmental, biological stress. The way to think about it is your skin cells are epithelial cells, protect your whole body. And when you go out there in the sun, you don't put sunblock on, what cells pay the price?